Now we will continue with the part which has been mentioned in the syllabus under reproductive system because it is related to that. So it is the part about genetics. So when we want to understand some concepts of genetics, there are certain ideas that we should understand first. The first thing is, what are chromosomes? So these are tiny thread-like structures which are found inside the nucleus of plant cells as well as animal cells. Now, in human beings, there are 23 pairs of chromosomes, and in these 23 pairs, 22 pairs are autosomes, and one pair is the sex chromosome. So, that pair, which is the sex chromosome, will decide or determine what is the gender of the person. Now, another term that is used in genetics is gene. So, when we say gene, what we mean is it is a segment or a particular part of the DNA molecule which is going to give the code for any particular characteristic of the organism. So, we know that all of us, we have the DNA inside our cells. So, on the DNA, the genes are located, and this DNA is there in the 23 pairs of chromosomes. So on that DNA double helix, whatever particular part segment which codes for a particular character is called as a gene. Now every gene has two alleles. Now these genes determine what will be the physical and genetic constitution of the organism. Now the next thing we need to understand is DNA. So we already know that DNA stands for deoxyribonucleic acid. We know that DNA has a double helix structure. There are two chains which are made up of multiple nucleotides arranged one after the other. And these two chains are wound around each other to give the double helix. And both these chains run in opposite directions. So they are called as anti-parallel. The other term is transcription. So when we say transcription, we mean the process by which the DNA will give a code, and from that we will synthesize the RNA. So synthesis of single-stranded RNA from DNA is called as transcription. So in this process, there is transfer of genetic information from DNA to RNA. The next process is translation. So here the RNA which is having the genetic information will convert that into a sequence of amino acids so that there can be synthesis of protein. So the process of protein synthesis by using the code which is on messenger RNA is called as translation. Then the central dogma of genetics is the theory which states that the flow of genetic information is only in one direction, and always it will be from DNA to mRNA, and from mRNA to protein synthesis. Now, what is important for us? All these terms are required for us to understand the concepts of genetics. So here what is important from our syllabus point of view is the genetic pattern of inheritance. That how do certain qualities pass from the parent to the offspring? So what is the inheritance pattern? What is it that by heredity, how do certain characters transfer themselves? Right? So the type of interaction which is there, that we have to see. So mainly this inheritance can take place in three ways or three methods are there. There is something called as incomplete dominance. The second method is called multiple allele inheritance, and the third one is called as sex linked inheritance. So what are the three methods of inheritance and examples for each of these we will see. So the first one we have to consider is the pattern called as incomplete dominance. So um, in certain plants, for example, there will be alleles which are going to be responsible for a particular trait. So depending on whether that allele is dominant type or recessive type, that quality will be expressed in a particular way, right? So if we take the example, of this plant, which is called as the 4 o'clock plant because it blooms at 4 o'clock in the evening. The biological name of that plant is Mirabilis jalapa. So suppose you take two pure parents of that 4 o'clock plant, okay, and you do a hybridization between them. So in this 4 o'clock plant, there are two types of flowers which are produced. There is red color flower plant and there is white colored flower plant. Okay. So if we take a red colored plant and in that we denote the alleles as capital R because both are the dominant type. So the both of them will have that dominant color which is the red color. Now if you cross that with a white flower plant, in the white flower plant it is the um, recessive trait which is there, which we will say because it is not red in color, it is white color. So it will be small r, small r, right? So the red colored plant is capital R, and the white flower plant is small r. So if you cross these two plants, okay, it is found that the F1 hybrid, F1 means the first generation hybrid, the offspring who are obtained by crossing these two varieties, that plant, the flowers will not have red color. You have crossed a red colored plant with a white colored plant, and you will find that the color of the flowers in this F1 hybrid is in between white and red. That is why those flowers will have a pink color. Now, if you take these pink colored flowers, that plant if you take, and you allow breeding within themselves, all the pink colored flower plants itself you allow to breed, then you will find that the next generation, that is the F2 generation. So in that generation, the plants may be of three different colors. They may be red, they may be pink, or they may be white. So the ratio of that will be one part of red, two parts of pink, and one part of white. So here, this kind of a pattern is called as incomplete dominance. So to explain, this picture here. So as we can see in the picture, this parent generation, we have taken a red colored flower, which is R1 they have denoted as. Then we have taken the white color flower, which is denoted as R2. So when you cross these two, right? Suppose you cross these two, that offspring, which is of the first generation, that hybrid will have a combination of R1 and R2, isn't it? Even either way, when you split this R1 into two separate units, it will be R1. That will be R2. So if there is R1 and R2 combining, and again R1 and R2 combining, whatever it is, your first generation will have all only R1 and R2. So that will be the pink colored flowers. So neither the white color flowers are there nor the red colored flowers are there, but the color of the flower is in between those two colors. So that is the pink colored variety. Now if you take this R1, R2 flowers itself, that generation of plants, and you allow them to breed between themselves. Now, all of them are the pink colored plants, pink colored flower plants. And if you allow them to breed between themselves, what can be the possibility? You can have, so one flower will be R1, R2. And you imagine here, I have not shown it in the box, but you imagine here there is another box with R1, R2, right? And what can happen? You can have four different possibilities. R1 of this flower and R1 of that flower can combine, or R2 of this flower and R1 of that flower can combine, right? Similarly, you can have R1 and R2 again combining, or you can have R2 and R2 combining. So if R1 combine, then you have this red colored flower. If R1 and R2 are produced, you have a pink colored flower. If in the second set R1 and R2 are produced, you have a pink color. And if it is R2 and R2 which are produced, then you have the white color. So here when you look at these four plants which are produced here, so one of them is the red variety, one of them is the white variety, and the remaining two are the pink variety. So that is why they are saying that the ratio in which the flower production will take place is one will be red, two will be pink, and one will be white. So this kind of an inheritance pattern is called as the incomplete dominance pattern. Then we have The next type of inheritance pattern is called as the multiple allele inheritance. So this we can understand by understanding the example of blood groups in human beings. So generally there is a pair of alleles which will control the phenotype expression, but in certain conditions for one character there may be three or more alleles. So this when this is the case, it is known as multiple allele inheritance. Now, in human beings, we know that there are four blood phenotypes which are present. You have people who have A group of blood, people who have B group blood, people who have AB group, and people who have O group, right? So, in blood group A, there is antigen A. In blood group B, there is antigen B. In blood group AB, there is both A as well as B antigen. And in blood group O, there is neither A nor B is present, right? So that means the inheritance will be based on these three alleles. We can denote it as IA, IB, and IO. Correct. So IA and IB are called as dominant alleles, whereas IO is the recessive allele. Right. Now this IA and IB can dominate over I0, but they cannot dominate over each other. So if you have IA and IB, then you will have both those A and B antigens. So this kind of a pattern is called as multiple allele inheritance. Now we can take a look at this diagram here. Now please note there's one diagram I had given in the notes, and in that example and what I have explained in the words is not matching. So that is a mistake that I have made. So please remove that diagram and instead you put this diagram into that part. So here we have the condition where the male is having a blood group A and the female is having the blood group B. So what will the children of the first generation, what can be their possible blood groups? So if you say the father is blood group A, that means let us consider that he is having IA as the dominant one and I0. I0 is also denoted as, IO is also denoted as a small I. Now the mother is having blood group B. So she will have IB and she will also have IO or small I. Small I is the O part of the antigen. So now when this father's gametes are produced, it can carry either IA or only I. The mother's B blood group, the gametes will be IB or only I. So depending on how they combine with each other. If the IA combines with the IB, then both IA and IB are dominant alleles. So both antigens A and B will be present. So that offspring, that child will have AB blood group. If the IA combines with the I of the mother, right? Then in that case, that small I is recessive and the IA is dominant. So then there is blood group A in that child. If the recessive gene of the father, small I, combines with IB of the mother, then the IB will be dominant. So IBI, small I, that is produced, and that means the blood group will be B blood group in that child. And if the I of the small I of the mother and the small I of the father combine, then that means there are no A and B antigens at all in that. So then that child will be having the blood group of O. So this is an example for what is called as multiple allele inheritance. So this you can try similarly with other options also. Suppose the father is AB and mother is B, or father is A and mother is AB. Like that, you can work out different options and you will find what are the possible blood groups which can be there in the children. So that is about multiple allele inheritance. Then the third type of inheritance pattern is when the inheritance occurs because that particular quality is transferred through the sex chromosome. So then that is called as sex-linked inheritance. So now we know that sex chromosomes, the XY or XS set, that is responsible for determining gender of the child. But also these sex chromosomes are responsible for some other non-sexual characters also. So many of the genes they have found that they are present on the X chromosome, but they are not there on the Y chromosome. So if they have to get transferred to the next generation, they will get transferred only when the X chromosome is transferred in that particular way. So this kind of pattern is called as sex-linked inheritance. And the most common example of that is the condition called as color blindness, red-green color blindness, where the person cannot make out the difference between red color and green color. So they will have deficiency of the cones in the eye. In the retina, there are rods and cone cells. So there will be a deficiency in the cones which are sensitive to red light or green light. So because of that, they cannot make out the difference between red and green, and both will appear the same color to them. So this gene for red-green color blindness is the recessive gene, and it will be designated as small c. Or and the gene for normal color vision will be capital C. Okay. Now these genes, whether it is a capital C or it is a small c, they are located only on the X chromosome. So they will not be found on the Y chromosome, right? So whether a person is able to see the colors or not depends on what kind of the X chromosome they are getting from the parent. So here we have shown in this picture one example of a person who is a male is normal. Okay, so for that normal man, if you see here, we are designating his genetic condition as XY, that X star Y, you can say. So the star near the X, that small asterisk mark, that indicates that it is a normal X chromosome. There is no gene for color blindness on that chromosome. And here, this woman, okay, let us say the man is not having the color blindness gene, but the woman is carrier. She is not having color blindness, but she is carrying the gene for color blindness. So that is indicated as XC, that superscript C is there, and the other chromosome is a normal chromosome, so X star. Okay, so now when the gametes are formed, one will be the XC gamete and one will be the X star gamete. In the normal man, one is the X star gamete and the other is the Y gamete. Correct? So now when the children are born, when the crossing happens in the first generation, now what can happen here? That if you have this XC combining with the X star of the man, the XC of the woman combines with the X star of the man, then the product, the offspring will have XC X star. So since that XC is carried here, this is a carrier daughter. Now here what will happen to the other daughter? This X star of the woman and the X star of the man. So X star and X star, that means there is no gene for color blindness there. So this X star will be a daughter who has normal vision. Now, what is the other option? This XC of the woman will combine with the Y of the man. So then the XY set indicates it is a son, it is a boy. And because that X chromosome is carrying that color blindness gene, the Y will not carry, we said. So the X is the one who is carrying it. So because that color blindness gene is there on the X, then this son who is born will be colorblind. And what is the other possibility? That the X star of the woman combines with the Y of the man. So the X star is the normal vision gene. So then that X star Y will be the normal, the son with the normal vision. So like this, different possibilities. Again, if you want to work out for your interest, you can consider where what will happen now if you have a color blind man crossing with a normal woman, or you have a color blind woman and a color blind man who are crossing. If you have a carrier woman and a colorblind man, when they have children, what will be the possibility? Like that, there are different scenarios which are present. So this kind of an inheritance pattern where the character is inherited or passed on to the next generation because of it is being present on the sex chromosomes. That is called as sex-linked inheritance. So with that, we have completed this part of the reproductive system. Please make sure again what I mentioned, that this picture for the A, B blood grouping, whatever I showed, the image which is there in the notes is not proper. So please make sure that you replace it with this example. Okay. Now we will go over to the other part. So now we will just consider some parts of the endocrine system, which maybe I should explain. the other part about the particular glands, then about their structure and about their function, you please study from the notes. Now we will just try, I will just cover some important parts of this endocrine system. So when we say endocrine system, we are talking about the system in which the hormones are synthesized and they help to maintain homeostasis in our body. So there are many different endocrine glands. And we've already studied about the difference between the endocrine gland and the exocrine glands. So we know that endocrine glands are the ones where the secretions are produced and there are no ducts for these glands. So directly they are released into the blood, and through the blood they travel to different parts of the body and they will act on particular organs and carry out their reactions. So we know that there are many important endocrine glands in the body. Pineal body is present, pituitary gland is there, thyroid gland, parathyroid gland, adrenal gland, pancreas, ovaries in females, and testes in males. So all these are the major endocrine glands. So the secretions of these endocrine glands are called as hormones. We define these hormones as chemical substances secreted by the endocrine gland which control specific physiological processes. So once the hormones are secreted, they will enter into the blood, and through the blood they will reach the target organ or tissue, and there they will exert whatever action is required. Now all these hormones, they have particular properties, similar ones. They have low molecular weight, they are soluble organic molecules, they have a high rate of diffusion, right? They are very effective in very low concentration. Even a small amount of the hormone is enough to bring about the desired action. They all travel through the blood. The site of action, the target site, is different from the site where they are produced. Like for example, we know that the pituitary gland is producing so many hormones, but they are not acting in the pituitary gland. They are going and acting on the testes, or they are going and acting on the adrenal glands. Like that, different areas of action will be there. So the action is specific to a particular target. That means every one particular hormone will act on a particular organ and it will exert one particular type of effect. Now, if you are having insulin which is getting released from the pancreas, now that insulin will act on particular type of cells and exert a particular type of action. Now, when anterior pituitary is releasing FSH, that action of FSH is separate. It is not the same as that of insulin, isn't it? So that is the meaning of saying that the action of the hormone is specific to a particular target, right? And they are non-specific for organisms. So if you have oxytocin, like I think I told in the class that day, that nowadays girls attain puberty faster because they are consuming milk which is obtained from the cow. Now in the cow, to increase milk production, they would have given oxytocin injections. So that oxytocin hormone which has been injected to the cow, not only it is having effect on the cow, but it will also get excreted out through the milk of the cow. So whoever is consuming that milk of the cow, that oxytocin comes through that milk and has an effect on that body of that person also. So the oxytocin was given to the cow. So that means the action of oxytocin is not specific. Whether it is on the cow, it will stimulate reproductive system. Whether it is in the human being also, it will stimulate reproductive system. So that is the meaning of saying that the actions of these hormones are non-specific for organisms. They are not specific that only in this organism they will have effect. In any organism, they will have similar kind of an effect, right? Then what are the functions of these hormones? Firstly, they are acting as regulatory and homeostatic in nature. So they will help to regulate the levels of certain items in our body. They will help to maintain homeostasis. They will help to maintain blood pressure. They will help to maintain body temperature. They will help to maintain a particular way of functioning. They will help to maintain particular level of calcium in the bones. Like that, they have functions. Then they will maintain consistency in the interior of the cell. Then they will permit movement of substances inside and out or outside of the cell. Most of the times they will have the effect of balancing between two systems, and they will, some of them have the function of helping in development of the fetus. So all this you need to understand to know about the hormones. But from the examination point of view, this part is not very important. What is important for us is the classification. So the classification of hormones is what is important from our point of view. So we can classify hormones in different ways. Sometimes we classify based on the chemical structure. They can be classified based on the mechanism of action. They can be classified based on the effects that they produced. Okay. So for us, what is important is classification of hormones based on the chemical structure. So we can classify into three categories. The first type of hormone are the ones which are protein in nature. So we call them as the peptide or protein hormones. So examples for this are the thyroid stimulating hormone TSH, adrenocorticotropic hormone ACTH, oxytocin, insulin, parathyroid hormone, calcitonin, and renin and angiotensin. All these are examples of hormones which have a peptide or protein kind of a structure. Then we have the second category, which are called as the steroid hormones. The second category of hormones are the steroid hormones. So we have hormones like cortisol, aldosterone, testosterone, estradiol, and progesterone. So all these are examples of steroid hormones. The third type of hormones are those which are derived from either amino acids or they are derived from fatty acids. So thyroxine, I'm sorry, here there's a mistake in this file. I don't know why this is happening. Thyroxine is T4, triiodothyronine is T3. Okay, both these hormones, epinephrine, norepinephrine, melatonin, serotonin, prostaglandins, all these are examples of hormones which are derived from amino acids or fatty acids. Now, we have to understand how these hormones are exerting their action. So, the mechanism of hormone action is important. Even in the exam, sometimes they ask about this. So, there are two ways in which hormones can act, and that depends on whether they are lipophilic or they are water soluble, hydrophilic. So I think you know the meaning of these two words. Lipophilic means they are insoluble in water, but they are soluble in lipids, right? And hydrophilic means they are soluble in water, but they are not soluble in organic solvents or in lipids. So if the hormone is a lipophilic hormone, now what happens is the cell membranes in our body, they are made up of lipopolysaccharide. So the lipophilic hormones can cross through the cell membranes, but the water soluble hormones cannot cross the cell membranes. So these lipophilic hormones, what happens is they cross the cell membrane, and after crossing the cell membrane, there are receptors which are there inside the cells. So the lipophilic hormones will bind, they will combine and attach to those intracellular receptors. So examples of such hormones are steroid hormones, thyroxine, and cholecalciferol. All these hormones are lipophilic. So they will cross the cell membrane, they will bind to the receptors inside the cell, and by that binding, they will regulate some process inside the cell. So whether it is a process of transcription, whether it is the production of specific proteins, right? You can either increase the level of some proteins or decrease the level of particular proteins. So that is what will be done by these lipophilic hormones. Then the next type of hormones are the water soluble hormones. Now these water soluble hormones, as we said, they cannot cross the cell membrane. So because they cannot cross the cell membrane, they will remain on the surface of the cell itself. So these water soluble hormones, because they are not lipophilic, they cannot cross the cell membrane. Because they can't cross the cell membrane, they will bind to whatever receptors are there on the outside of the cell, on the surface of the cell. Now, when this hormone binds to the cell surface receptor, it will make some kind of a change inside the cell. And inside the cell, there will be release of certain compounds which will carry the message, what has been brought by the hormone. So those compounds which are released inside the cell are called as secondary messengers. And because of the release of the secondary messengers, there will be certain functions which happen inside the cell. So hormones like ACTH, FSH, insulin, and prolactin. These are examples of water soluble hormones, right? So both these mechanisms are possible. So if you are asked to explain about the mechanism of hormone action, you have to talk about how hormones can be lipophilic or they can be water soluble. And if they are lipophilic, what is their mechanism of action? If they are water soluble, then what is the mechanism of action? So in this water soluble hormones, we said that when the hormone binds to the receptors on the outer surface of the cell, that will stimulate secondary messengers, right? So that hormone which comes and attaches to the receptors is called the first messenger, and the other compound which is stimulated inside the cell is called as the secondary messenger. So there are many important secondary messengers in the body, inside the cells, like cyclic AMP. I think you know the full form of AMP, adenosine monophosphate. So the cyclic AMP is written as small c AMP. Then protein kinase is also an example of a secondary messenger. Cyclic GMP, guanosine monophosphate. Cyclic guanosine monophosphate is also a secondary messenger. And phosphoinositides. These compounds also act as secondary messengers. So all these come under the mechanism of hormone action. Now, the remaining part of that endocrine system that you will study from the notes itself anyway. So, it is just about every particular gland, what is the structure of the gland, what is the function of the gland, and what are the disorders related to that particular gland. So, you please study that from the notes. If you have any doubts at any point of time, you can message me and ask me. Now, here there is another thing which I had not included in the first part of the notes of endocrine system, and I sent it separately. I hope you have looked at that also. So here there are two important things. These are asked also in the exam many times. So we need to understand about what is called as hormone feedback mechanism. That is one thing. And the other part is what is the role of insulin in our blood glucose maintenance. So first we will take up about this hormone feedback mechanism, right? Now we said that the role of the hormones is that they control homeostasis. So the hormones are playing an important role. Then how does the control of the hormone take place? That is regulated by a mechanism called as a feedback mechanism. So generally when we say feedback, it means that something has been done, and at the end of that process something is going back and trying to increase or decrease that mechanism itself. So a feedback mechanism is defined as a loop in which a product feeds back to control its own production. So what will be happening is you have a reaction which is taking place. Some particular compounds are reacting together and giving one particular product. When the level of that product increases more, that product will go back and it will prevent the reaction which was producing that product. So that is actually what is a feedback mechanism. Now here, when we have feedback mechanisms, we can have two types. One is called as a positive feedback mechanism and the other is called as a negative feedback mechanism. So most of the times in hormone feedback mechanism, it is the negative feedback mechanism which is taking place. So what exactly happens in this negative feedback, right? So if the level of a particular hormone increases, now the level will go on increasing. Now after some time, you don't want the level to increase beyond that particular level because it is not going to be of any use. So at that point, or it may have some harmful effect also. So that then what has to happen? There has to be some mechanism by which the further production of that hormone will stop because now sufficient level has been attained. So that kind of a feedback is called as negative feedback. So one very good example for this negative feedback mechanism in hormones is the thyroid gland. So what happens? To understand how this negative feedback is working, we have to see how actually the production of the thyroid hormone takes place. We have studied already about the synthesis of the thyroid hormone. Then we also said that whenever there is a signal from TSH, from the thyroid stimulating hormone, then this T3 and T4 will get released, isn't it? So what happens is in the brain, the part of the brain called the hypothalamus, it secretes a hormone called as the thyrotropin releasing hormone. This thyrotropin releasing hormone is sometimes also called as the thyroid regulating hormone. So that stimulates the pituitary gland, and from pituitary gland, TSH, thyroid stimulating hormone, is released. Right. Now this TSH, it will come through the blood and it will reach the thyroid gland. And when the TSH acts on the thyroid gland, then the thyroid gland will increase the secretion and the release, the production and release of the T3 and T4, triiodothyronine and thyroxine. Now, after some, as this is going on and on, what is going to happen now? The level of T3 and T4 starts rising. So, once the desired level is reached, we don't want further production of T3 and T4. What will happen now is this T3 and T4 itself will give the signal to the hypothalamus. When the blood level of T3 and T4 becomes very high, that blood which is reaching the hypothalamus, it will get the signal that now there is enough T3 and T4 which is present. So it will signal the hypothalamus to stop the secretion of TRH. When TRH secretion stops, then the pituitary stops producing TSH. So when TSH stops, then the stimulation of the thyroid hormone also stops. So the production of T3 and T4 will stop. Now whatever T3 and T4 is there in the blood will get used up. After some time, what will happen now? Again, the level of T3 and T4 will come down. So again, the cycle will start. So this is what is called as the negative feedback. So here in the diagram, if you see, they have shown it that we have this TRH, the hypothalamus is here, the pituitary gland is in the middle, and the thyroid gland is there at the bottom. So this hypothalamus will release the TRH. That TRH will act on the pituitary gland. It will cause release of TSH. That TSH is coming to the thyroid gland and it is stimulating the thyroid gland to produce T4 and T3, the thyroid hormones, right? So all this part you can see it is shown in green. That means it is stimulating the production. So TRH is stimulating production of TSH. TSH is stimulating production of the thyroid hormones. Now the thyroid hormone level in the blood will go on increasing. Now once the level of thyroid hormones increases very high, then we don't want more production of T3 and T4. So this, you can see this line which is going backwards and in red color. So what this T3 and T4 will do, this minus sign, you can see it here. So they are having a negative feedback they are giving to the TSH. They are also giving negative feedback to TRH. So the T3 and T4 will go to the thyroid regulating hormone and prevent it from stimulating the pituitary gland. They will also go to the pituitary gland and prevent the secretion of TSH. So because of that, what will happen? The thyroid gland stimulation will stop. So when thyroid gland stimulation stops, then production of the T3 and T4 hormones will stop. So then what will happen? Whatever T3 and T4 are there in the blood, they will be utilized for whatever action is required in the body. Now, after they are completely utilized, what is going to happen now? The level will start dropping. So when the level starts dropping, then this negative feedback will stop working. So when negative feedback stop working, again that cycle starting from the beginning. TRH is produced, it will stimulate pituitary, it will stimulate thyroid gland, again thyroid will be produced. Again the levels will rise to a particular level, those will go and inhibit the TRH and TSH, and because of that production will stop. So this is what is called as a negative feedback loop. Okay. Now positive feedback. So in negative feedback loop, we saw that there is the product of a particular reaction and that product will inhibit its own synthesis when the levels become very high. In positive feedback, what happens is as more and more product is being produced, then there is more and more production of that same product. So that is why it is called as a positive feedback. Increasing quantity of the product will increase more synthesis of that particular product. In negative feedback, what we saw, increasing quantity of the product is going to decrease the production of that product. But in positive feedback, what we see is as more and more product is produced, more and more its production will increase. So an example for this kind of mechanism is the production of milk by the mother for the baby. Now what happens after childbirth? We know that oxytocin starts stimulating the release of milk from the mammary glands. As the baby is sucking the mother's breast, the milk will start coming out. So as the suckling increases, there are receptors in the mammary glands, in the nipple. So those receptors will carry the message to the pituitary gland. That pituitary gland will secrete the hormone called prolactin. Now this prolactin will come and stimulate the mammary gland to produce more milk. So the baby, now as more milk is being produced, the baby will drink more milk. So as the milk is drunk more and more stimulation of pituitary will take place. Again, more prolactin is produced. Again, more milk production is there. Now, this is important for us, isn't it? Because if it was working, if this mechanism had to work by negative feedback, then you imagine what will happen. More milk will be produced. Then the baby will drink the milk. Then after that, what is going to happen? The milk will stop produced. That is going to be damaging, isn't it? Because we want the milk to be available at all times for the baby to get nutrition. So this is positive feedback mechanism, which is very useful for us. So more drinking by the child, more production of milk by the mammary glands. So this is the positive feedback mechanism. So these two mechanisms are possible. Now most hormones in our body, they are acting by negative feedback mechanism. And only this milk production is an example of a positive feedback mechanism, right? Okay. Then in the questions paper somewhere, they have asked a question about how blood glucose is controlled by using insulin and glucagon in our body. Okay. So we know that insulin and glucagon, they are hormones which are produced by the pancreas, right? The alpha cells of the islets of Langerhans produce glucagon, and the beta cells of the islets of Langerhans in the pancreas, they produce insulin. Now, when a person is consuming carbohydrates in the diet, right? Whatever carbohydrate, ultimately it will be starch, and that starch will get converted in our body into glucose, right? Now, the body cannot use all of that glucose. So, some of that glucose will get converted into glycogen, and that glycogen will be stored in the liver and in the muscles. So, whenever the body requires more energy, and if the glucose which is there in the blood is not sufficient, then the body will break down that glycogen which is there in the liver and convert that glycogen into glucose. So in this way, the balance will be maintained. So what happens exactly? Now, when a person has a condition of high blood sugar level, that is called as hyperglycemia. Now, if somebody suffers from hyperglycemia and too much high level of blood sugar is there, then it will cause lot of damage to many organs in the body. The kidneys can get damaged, the retina of the eye can get affected, then the immune system will get affected. So many, the heart will get affected. So many effects will be produced. So people who have diabetes mellitus, they suffer from hyperglycemia. So the opposite of hyperglycemia is hypoglycemia. Hypo means less. So hypoglycemia means low blood sugar level. So when somebody's blood sugar level goes very low, they will have dizziness, they will faint, they may become giddy, and in some cases the person may go into coma and they may even die if the blood sugar goes very low. So now our body has to maintain blood sugar in a proper balance. It should not become high also, it should not become low also. So for maintaining this balance, there is a what you say combined action of both insulin and glucagon. Right. Now let us assume that somebody has eaten carbohydrate rich diet and there's a lot of glucose which is produced in the body. Now that glucose has to enter into the cells. So for the glucose to enter into the cells, it is the hormone insulin which is going to act. Now you remember we said insulin is the water soluble type of hormone. So it will not go and go through the cells, but it will attach to the receptors on the cell surface. So when the body senses that there is more amount of glucose in the blood and that blood has to enter into the cells, then the pancreas will release insulin. So generally it happens that sometime after we take our food, the pancreas will release insulin. Now what is the role of this insulin? It will cause the cells to take up that glucose from the blood. So when the blood glucose enters into the cells, then the level of glucose in the blood will come back to normal. Now these glucose molecules have entered inside the cells of the body. So they are not there in the blood. Because of that, the blood glucose level will come down and it will become normal. Right. Now, what happens is that suppose there is still, now cells have taken up some glucose, but still if there is more amount of glucose, what will happen next is that the insulin will signal to the liver to absorb that glucose. So the liver will absorb the glucose and it cannot keep the glucose as glucose. It will convert the glucose into glycogen, and that process of synthesis of glycogen in the body is called as glycogenesis. I think you've studied this in biochemistry. So the glycogen will get converted into the glucose will get converted into glycogen and it will remain in the liver. Okay. Now this person's blood sugar has become normal. Now after some time, as the cells use up more and more of glucose, the blood sugar level, then the time has passed since the last meal what you have taken. Let us say some 4 hours pass, 5 hours pass, say 10 hours pass, and still you don't eat anything. Now what is going to happen to the level of sugar in the blood? It will start becoming low. So the body will sense that there is hypoglycemia, and we said hypoglycemia is a dangerous condition. So when that hypoglycemia starts, the pancreas will get the signal. So now what the pancreas will do is they will stop the insulin from getting released, and they will start in the pancreas will start increasing the release of glucagon. Now the role of glucagon is that it will increase the blood sugar level. So what the glucagon will do, it will send a signal to the liver, and it will tell the liver to convert the glycogen back into glucose. So this process of breakdown of glycogen into glucose is called as glycogenolysis. So because of that, what will happen now? The glycogen is converted into glucose and released into the blood. So the blood sugar, which had become very low, will now start to rise, and it will become to the normal level. At the same time, this glucagon can also stimulate another process in the liver, which is called as gluconeogenesis. I think you remember the meaning of this, synthesis of glucose from non-carbohydrate sources. So that way also there can be increased synthesis of glucose, so that the blood sugar level will now come back to normal. It had become too low. So when glycogenesis takes place, when glycogenolysis takes place, and when gluconeogenesis takes place, that blood sugar level will come back to the normal level. So that is what is shown here in this diagram. You have blood glucose low, then the pancreas will get activated. The alpha cells of the pancreas will release glucagon, and because of that, there will be glycogenolysis in the liver. So the glucose will get released into the blood, and that low blood glucose will become normal blood glucose. Similarly, at a time when there is high blood glucose, then that signal goes to the pancreas. The pancreas beta cells will release insulin, and because of insulin release, the cells will take up the glucose from the blood, and the normal blood glucose will come. Also, it is not shown in this diagram here, but these insulin cells will also promote the uptake of glucose by the liver. The liver will convert the glucose into glycogen and store it there, and because of that, the blood glucose level will become normal. So this is the mechanism. By using glucagon and insulin, the body will regulate the levels of blood glucose. So that is what, if you are asked to explain how insulin helps to maintain blood sugar level, or how glucagon and insulin work together to maintain blood sugar level, then you have to explain about this. You have to tell what is hyperglycemia, what is hypoglycemia, what happens when the blood sugar becomes less, and what happens when the blood sugar becomes more. So that is what has to be explained. You can draw this diagram also and explain. Fine. So that is all that was pending from my side for the explanations. That endocrine system, whatever other things are there, you please study on your own. At any point of time, if you have any doubts or you want to ask something, you can send me a message on WhatsApp. I will definitely clarify whatever it is. Okay. So study well, and all the best for your exams. Thank you.